Thank you. Thank you. Thank you, Liesel, for this introduction. First of all, I would like to thank the organization to giving me the opportunity to talk today about probably one of the most important phytopathological problems, not only in wine grapes, but also probably in table grapes, which is grapevine trunk diseases. So I would like to start my presentation giving you some numbers about the economic impact of these trunk diseases worldwide. Here you can see some examples. For example, in Australia, about 40% of the cultivar Syrah has some symptoms of Eutypa diabac, one of the main diseases associated with mature plants, causing an economic impact of almost 3,000 Australian dollars per hectare. In California, these diseases, Botryopsferia and Eutypa diabac, cause an economic impact of 260 million American dollars per year, and 12% of the French vineyards are affected by ESCA, one of the most characteristic diseases in mature plants, causing an economic impact of 1 billion euro per year. In Spain, in our region, northern Spain, La Rioja, we recently estimated the cost of replanting 1% of plants affected by grapevine trunk diseases, plants cultivar tempranillo, and the economic impact is about 7 million euros per year without taking into account the losses in wine production until the full yield. So we estimated 1% of these plants affected by GTDs, but sometimes in some vineyards we have 5, 8, and even 10% of plants affected by this pathosystem. There are several trunk diseases associated with these symptoms or with this pathosystem. Here you can see some examples of external symptoms. To the top left, we can see Blackfoot and Petri diseases. These diseases affect mainly planting material in nurseries and young vineyards. You can see the typical symptoms: stunted growth, reduced vigor, and sometimes wilting and dieback. To the top right, we have Botryopsferia diabac, typical symptoms of death of spurs and arms. Then Eutypa diabac, yellowish, chlorosis, shortened internodes, and finally, to the bottom right, we have ESCA disease, the characteristic decoloration in leaves, the tiger stripe symptoms, and suddenly we have the death of the plant, the sudden death of the plant during summertime. If we have a look at the internal symptoms, we have a broad range of internal symptoms, so necrosis at the base of the rootstock, sectorial and central necrosis, black spots in the xylem vessels, as you can see at the bottom left of this slide, and then also white rot. So obviously, this type of internal symptoms can compromise the translocation of nutrient and water from the roots to the aerial part of the plant. So this pathosystem is really complex. Usually we talk about powdery and downy mildews, one causal agent associated with one disease. Here we have more than 100 species belonging to 35 genera. We have diseases affecting nursery and planting material, young vineyards, other diseases affecting mature vineyards, and we have others, such as Botryopsferia and Phomosis diabacs, that are able to infect the plants in all the scenarios. The second factor that explain this complexity is that these fungi have different biology and epidemiology. I mean, they can live in different places, they can be spread by different ways. Several diseases or several pathogens live in soil and can infect the plant through the roots. Other pathogens can be spread aerially and infect the plant through the pruning wounds, and other diseases are specialized to infect the planting material in nurseries during the propagation process. And finally, the third factor that explains the complexity of this pathosystem is that, probably the most important, no curative measures are known for the control of GTDs. So we have to use, as Liesel mentioned before, an integrated management strategy, including different tools, given less priority to the chemical methods, as you will see in my presentation. So this integrated management strategy should be applied in all lifespan of the vine, of the plants, from nursery mother blocks through the propagation process until the mature vineyards. So in this first part of the presentation, I'm going to focus in nursery monoblocks and mature vineyards, giving priority to the studies published under field conditions. So the rootstock model vine cultivation can have an influence on grapevine trunk disease infection. So here you have some examples of the different ways to cultivate, in this case, rootstock model vines. For example, at the top left, we have in Spain, which is quite common to cultivate the rootstocks sprawl on the soil surface. This can be detrimental because, you know, the planting material is in contact with the soil and we can get cross-contamination by soil-borne fungal pathogens. In South Africa, for example, I saw this strategy some times ago. They use like a woven fabric just to avoid planting material to be in contact with soil, which can be quite interesting. But for me, in my opinion, I think the best strategy is to use the trellis systems. Trellis systems can provide some agronomical advantages compared to the other systems and also avoid this soil surface pathogen contamination or potential soil surface pathogen contamination. It's clear that probably this system is much more labor-intensive and expensive compared to the rootstock sprawl on the soil surface. Irrigation can also have an impact, an effect on grapevine trunk disease infection. For example, I strongly recommend the drip irrigation. Here I show you, in the case at the bottom right of this slide, the flat irrigation. This is quite detrimental. You know, the overwatering favors most soil-borne pathogen infection, so this should be avoided. And another type of irrigation can be the sprinkler irrigation, but this system can trigger spore release of several GTD fungi, for example, Botrytis, Phagramonium, and the Atripasi species. So this method, in terms of phytopathology, should be strongly avoided. In mature vineyards, it's important to understand the cycle of these diseases in order to know when we should apply the different strategies to minimize fungal infection. Here in this slide, I show you the disease cycle of Botrytis dieback. In general, the cycles of these diseases affecting mature plants are quite similar. There are some changes in the fruiting bodies and the type of conidia, but in general, they work quite similar. So if we have a look at the top of this disease cycle from the pruning debris and the infected dead wood, this pathogen can produce fruiting bodies, resistant structures, especially during winter time. Then in spring, when they have like adequate temperature and humidity, these spores are released from these fruiting structures. Then the spores are disseminated by wind, arthropods, or rain. They reach like a susceptible pruning wound. The spores germinate and cause the internal cankers or internal symptoms. Then after some time, we don't know exactly if it's several weeks or even years, we see the external symptoms. So the disease cycle is closed. One way to stop the disease in this case will be to remove pruning debris or infected dead wood. So actually, the dead wood or the pruning debris are potential source of inoculum of GTD pathogens. So in this study, researchers in northeast Spain left pruning debris in the vineyard for several times after pruning, and they observed that 42 months after pruning, these pathogens were still able to produce fruiting structures and viable conidia. So it's important in this case reduce this inoculum source, eliminating the pruning debris. Here's some example in table grape in Sonora, Mexico. I took this photo last February. The pruning debris can be an inoculum source of these pathogens and provide like secondary infections. There are several ways to remove the pruning debris in this case. So probably the most effective would be burning this planting material, but it has some environmental disadvantages, and this is not allowed in many regions around the world. So there are other strategies. I mean, this strategy has been replied by others. For example, like grinding and leaving the planting material on the ground, such like a mulching, or even grinding and incorporating this planting material into the soil as an external or additional supply of organic matter. We don't know exactly scientifically if this strategies can eliminate completely the fruiting bodies and the spores, given the small size of the fruiting bodies, 500 micrometers, or the spores around 10 micrometers. So it would be nice to evaluate if these strategies have some effect in terms of phytopathology. And probably the most interesting and sustainable strategy will be composting. In this study, French researchers at INRAE in Bordeaux, they mixed the pruning debris together with sheep manure and garden residues and got percentages of 0% of infection by different species associated with grapevine trunk diseases. So composting would be a nice strategy to use in this case. It's not available for all wine growers or all table grape growers, and probably is a little bit more expensive than the other strategies. So let's think that it's almost impossible or is really difficult to eliminate, to remove all the pruning debris and infected dead wood from our vineyards. So the pathogen will continue the disease cycle, and an additional factor that makes this pathosystem more complex is that these pathogens can also infect alternative woody hosts. Here you have some example of GTDs affecting, or trunk diseases affecting almond, pear, and walnut. This is, in this photo, you can see the typical scenario of a Mediterranean region where grapevine is planted along with other crops, peach, almond, olives, etc. So there is a hypothesis that says that maybe there is an exchange of inocula between grapevine and these woody hosts. So this is probably adds complexity to this pathosystem. So the pathogen will continue, the spores will be released from these fruiting structures, produced on grapes and woody hosts, and then we will get into the pruning wounds. So the annual wounds, mainly made during winter, are, as Akif said before, is probably one of the main point of entry of these pathogens into the plant. So you can see here a couple of examples of the number of wounds made in the mother vines, in the nurseries, and also in mature vines in the vineyard. So much research has been done in evaluating different products to, in this case, to protect pruning wounds. And I mean, in several countries, Australia, South Africa, US, Chile, and some of them, or most of them, concluded that the mastic or paste plus fungicides belonging to the benzimidazole carbamate mode of action group or the dimethylation inhibitors are the most effective in protecting pruning wounds against grapevine trunk disease pathogens. Recently, a couple of papers published in California and Spain demonstrated that this mixture, pyraclostrobin plus boscalid, was very effective in protecting pruning wounds. However, as mentioned this morning, in one of the presentations, now we have huge restrictions on the use of chemicals in viticulture, especially in the European Union. So we need to find, we need to find alternatives. And there is now a frantic search for this alternative. One of them is the use of biological control agents, especially different species of the genus Trichoderma. Lisel already highlighted the mechanism of action of this Trichoderma species. Trichoderma, under my point of view, is really effective under control or semi-controlled conditions, laboratory, greenhouse, nurseries, but under field conditions, we saw many variable, inconsistent results for preventing infection by GTD pathogens. And the efficacy of these products depend on the grapevine growing region, climatic conditions, the genotype, and the experimental design. I mean, if we use artificial pathogen inoculation or natural infection. We, as plant pathologists, used to inoculate artificially plant pathogens, putting high inoculum pressure in the vineyard just to get good statistical analysis and good differences with respect to the untreated control. However, this doesn't happen in reality, in nature. I'm going to show you a couple of examples of these variable results when using Trichoderma species. The first one is the use of this mixture, Trichoderma sperellum ICC12 and Trichoderma gamsii ICC80. So under artificial pathogen infection, under high inoculum pressure, this product, this mixture of two trichodermas, worked very well in Italy, as Liesel mentioned, so 9-year trial, reducing the tiger stripe symptoms from 66 to almost 90%, and also in California, research done by the group of Akif, this trichoderma species performed the best against two fungal pathogens associated with grapevine plant diseases. However, under natural pathogen infection, surprisingly, under lower disease pressure, we evaluated this product in Spain and France for 3 years, different genotypes, different locations, different climatic conditions, and this product was the worst, yielded inconsistent results, which were related to the year, the location, and the specific disease in nature. Another example, Trichoderma atroviridae I-1237, the commercial product is Esquif. Under artificial pathogen infection in Galicia, northwest Spain, this product did not reduce the infection by Diplodia seriata. Diplodia seriata is one of the main causal agents of Botryosphaeria diebac. However, under natural pathogen infection in another region in Spain, in La Rioja, northern Spain. So this species of trichoderma was the most effective, reducing infection by different Botryosphaeriaceae species that were living in nature. In South Africa, you have two products. The first one is a chemical product, hydroxyquinoline sulfate. We evaluated this product in 2009 under in vitro conditions, and we demonstrated that this product is not effective in eliminating fungal trunk pathogens. The other one is Trichoderma atroviridae, the ICO77, which is registered specifically against U-type palata. So there are several studies using this trichoderma. In South Africa, for example, under artificial pathogen infection, different chemicals, the benzimidazoles, were superior to trichoderma, but trichoderma ICO77 caused a significant reduction in U-type palata infection. U-type palata is the main causal agent of U-type diebac. And also, under natural pathogen infection, this species of Trichoderma atroviridae not only reduced U-type palata growth, but also the incidence of different grapevine plant disease pathogens living in nature. In Canada, they also use this species, Trichoderma atroviridae ICO77. This is a paper which is now online in the American Journal of Enology and Viticulture, and the researchers show that this ICO77 provided effective pruning wound protection against different species of Botryosphaeriaceae for up to 60 days. One of the issues is the cost of treatment. So in general, research showed that the manual painting with fungicide is much more effective than spraying the product. However, this strategy is much more expensive, 3 and even 4 times more expensive compared to spraying fungicides or biological control agents, as shown in this Australian study. Another issue is when should we apply the protective treatments. So I strongly recommend to protect the Trichoderma species or the biological control agent immediately after pruning, because these fungi have to germinate, establish, and grow just to protect the pruning wound against different species. And regarding chemicals, in this Australian study, the researchers demonstrated that you can apply these active ingredients, tebuconazole, fluazinam, and pyraclostrobin, up to 6 days after pruning, so the efficacy is similar. So this can give some flexibility to the grower compared to the application of biological control agents. Once the plant is infected, so there is no curative treatment, so this is the last option we have if we have infection in our vineyard. So this is the remedial surgery. This is not new. I mean, in the European manuals of viticulture, early 20th century, we already saw this strategy. And it consists in removing visibly infected parts of the vines until getting apparently healthy wood, then trying to cover this wound by a mastic or paste and trying to renew the plant by using basal shoots. So the efficacy is not 100% guaranteed, and this is specifically indicated for local diseases such as Boytrospheria and Eutypa diabac. For example, in the case of ESCA disease, what we saw is that the necrosis can also reach the rootstock part of the plant, so below ground. So it's almost impossible to remove all this affected wood when we have plants affected by ESCA disease. Okay, so now the second part of my presentation, I'm going to focus in the propagation process, nursery field, and newly established vineyard. So unfortunately, nurseries provide nearly optimal environments for fungal development and humid conditions, so wet and humid conditions in hydration tanks. Also, the high root density and close spacing of plants in the causing stage and also the rooting stage. Even with these favorable conditions for disease development risk, the disease will not develop if the pathogens are not present. So, however, the production practices in box nurseries provide many opportunities for fungal infection. So here we can see the pruning wounds in rootstock and scion mother blocks, the disbudding stage, and also the dramatic action of putting together the rootstock and the scion. So another factor to take into consideration is that diseased plants are really difficult to detect. We usually see this type of plants in the nursery, like appropriate thickness of the rootstock, appropriate aerial part of the plant. But when we cut these plants, we see these internal symptoms like black spots, like necrosis, from which we isolate vascular pathogens, especially Pheomoniella chlamydospora and Phaeochromonium species. So it is thought that these pathogens are latent pathogens living in the xylem vessels, causing asymptomatic infections, and they become pathogenic following biotic and or abiotic stress factors. So if we want to see if we have a plant infected by grapevine plant diseases, we have to perform a destructive sampling. So this is the message we give to the nurseries. The slogan, start clean, keep it clean. Try to use pathogen-free planting material, which is not easy at all. Try to clean grafting machines frequently, maintain a high standard of general cleanliness in the nursery, particularly the grafting, causing, and cool rooms. Try to never reuse causing media, which is a potential inoculum source of these pathogens. Now nurseries are using water with copper, which is much better than peat or other causing media. And the last one is try to avoid soaking cuttings for long periods of time with water. Hydration tanks are also a potential inoculum source of these pathogens. So during the propagation process, chemical control again is almost forbidden in many countries around the world. Here in South Africa, you still have available the spore kill. This is an agricultural disinfectant, plant sanitizer. The active ingredient is the diethyl dimethyl ammonium chloride. And several research in South Africa and Spain demonstrated the efficacy of a spore kill against grapevine plant disease pathogens in nursery. But, you know, in most countries, we don't have authorized and efficacious products, chemical products, against these diseases to use in nurseries. So an alternative is the biological control again. Biological control mainly by Trichoderma species. Trichoderma species grow very well under semi-controlled conditions, I would say in nurseries, or controlled conditions. And this is one of the strains of the species Trichoderma turbidide, the strain SC1, that work very well against trunk diseases in nurseries. This research was done in Italy and Spain. Another strategy is the use of hot water treatment. So the standard protocol is 50 degrees for 30 minutes. These treatments have been widely used against several pests and diseases, especially phytoplasma organisms, but we have to take into consideration different factors. The first one is that some Vitis vinifera varieties are more sensitive to hot water treatment than others, so the viability can be compromised after these treatments. Second factor is that the tolerance of plants to the treatment is affected by the climate in which the cuttings are grown. For example, in New Zealand, they cannot treat of up to 48 degrees, but in Spain, we have like a warmer climate, and we can treat of up to 53 degrees. And the last factor is that hot water treatment is not completely effective in eliminating fungal trunk disease pathogen growth. So as in the case of biological control, these strategies reduce infection but not eliminate, in this case, the disease. Regarding disease resistance, the use of roostocks, so many research have been done in New Zealand, Spain, US, but none of the roostocks evaluated have shown complete resistance to Blackfoot and Petri diseases. Just some roostocks were tolerant. In the case of New Zealand, this roostocks handled 114 milliardet de Grasse to one specific pathogen, and in Spain we demonstrated that the roostocks 169-49 Cuder was tolerant to 7 vascular pathogens associated with Petri disease. However, just curiosity, this rooststock, the 161-49 Cuder, causes telosis, physiological problems, incompatibilities with some Gramaje clones, and also it shows poor behavior in compacted and wet soil. So in France, this rooststock has been removed from the market, and according to the French researcher, they call it like 161-49 decline. Here you can see some of the symptoms, so decrease in vigor, trunk diameter variation, so thick and non-adhesive phloem, and they also observe very small amount of starch in roots. Regarding the cultivar resistance, there are also a lot of research done in the literature. I'm going to show this one in California. So there is no single cultivar able to tolerate all the broad range of fungal pathogens. For example, in this case, we see that Concord was tolerant to Neophysicoccus parvum, one of the male causal agents of Botrytis diebac, but very susceptible to Pheumoniella chlamydospora, causal agent of ESCA disease. So some research pointed out that there is a relationship between the mean diameter of the xylem and the susceptibility to vascular pathogen infection. In the case of Thomson seedless, for example, Thomson seedless has a wider xylem diameter compared to other wine varieties and is much more susceptible to fungal infection. Next step is the nursery field. The nursery field is a potential inoculum source of soil-borne pathogens, specifically Blackfoot disease pathogens. Here you can see the disease cycle of these pathogens, which is quite simple. Blackfoot can infect grapevines via conidia. They can also produce resting spores, chlamydospores, that allow them for survival for a long period of time. Blackfoot can also infect alternative hosts during the rotation system in nursery fields, such as wheat or cereals. For example, in Portugal and South Africa, researchers demonstrated that an increase of Blackfoot disease incidence following this crop rotation strategy. Also, we detected these pathogens during the rotation system with wheat and barley. So nowadays, strong reduction on the use of chemicals. So again, chemical soil fumigation is forbidden in many countries, no authorized products. So one alternative could be the biofumigation. Biofumigation using Indian mustard seed meal proved to be effective in New Zealand to reduce Blackfoot disease incidence and fungal inoculum in soil. We also observed similar effects by using white mustard in Canada and Spain, Sinapis alba, in reducing this fungal inoculum. Pre-planting treatments, again, we should use biological control, no chemicals are available. So this research done by Lisel and Francois demonstrated that the use of different trichoderma species were not sufficient to prevent infection by Blackfoot disease pathogens, but a certain degree of protection was obtained in the basal end of the rooststocks. In Spain, we evaluated different biological control products or agents, bacteria, oomycetes, and fungal species associated with trichoderma. And what we observed is that a combination of more than 2 biological control agents were required to reduce fungal infection. And one important thing is that the survival of these biological control agents after 1 and 2 years after the application was very low. I mean, so the soil is a complex niche, where multiple interactions occur, so it's really difficult that these biological control agents under natural conditions grow well, establish themselves, and can compete against these pathogens. Another interesting issue is the use of mycorrhiza. Mycorrhiza improve nutrient acquisition, reduce water loss, and decrease plant resistance to abiotic and biotic stresses. However, regarding phytopathology, regarding trunk disease pathogens, so in Canada, they work a lot with native mycorrhiza, and they demonstrated that this native mycorrhiza provided low level of establishment in the vineyard, so the same as we observe in Spain by using our biological control agents, the inefficacy to prevent blackfoot disease infection, and also a lack of significant difference in agronomical features between the mycorrhiza inoculated plants and non-inoculated plants. So further research is needed using other mycorrhiza, native mycorrhiza in other scenarios to test against grapevine trunk diseases. And finally, I would like to talk very briefly about the management of young vineyards. So this is, in theory, for wine grape production. I assume that the table grape production is really different, but we theoretically recommend avoiding production the first 3 years and then increasing the production until full production 8 or 10 years after planting, according or depending on the rootstock and the scion combination. So there is a hypothesis that says that the stress factors, such as overcropping, can be related to disease development and symptom expression regarding grapevine trunk diseases. So we should avoid this overcropping. A plant which is stressed is more susceptible to fungal infection, not only grapevine trunk diseases. So this is my last slide of the presentation. I was compiling over the last 30 years all the influencing factors in symptom expression, grapevine trunk disease symptom expression, the research in the literature. So you can see here all the different factors that can explain the disease symptoms on grapevines. So the type of wounds, outer inoculum, foliar symptoms, climate, soil, nitrogen, fertility, many factors. And these factors are correlating, are making interactions. So you can see here how difficult it is to study, to do research on grapevine trunk diseases. Okay, so that's all. Thanks for your attention. I will be happy to take any questions you may have.
Thank you, David, for a very comprehensive talk, giving us a nice overview of grapevine trunk diseases in the nurseries and in the vineyard. We've got time for one question. Right, just one here. I don't know where the roaming mic is, over here.
Hola, David.
Hola.
Question. Is true that if after the use trichoderma in the field, you have to avoid spray another fungicide for a minimal period of time, 5 days, 7 days, I don't know how many, because you could kill the trichoderma with another conventional fungicide?
Well, you have to check probably the company, because each company says something different, but it's quite evident that some chemicals can kill these pathogens, this fungi. It's quite evident. So, but I recommend you just to consult, just to talk to the company, because some of them are telling you different things. This is big. I mean, I was asking the same as you are asking me now, and I got the same response. So just because some of them are telling you, okay, no problem, you can use this benzimidazole, you can use this, but some of them are telling you, okay, be careful and try to wait 1 week or 2 weeks before spraying with these chemicals. So I don't know if Liselle has any opinion about that.
Some chemicals are compatible, but others not. You really have to look at the specific chemistry. But in terms of trunk diseases applying during dormancy, I don't see the need for another fungicide. I'm not sure if your trichoderma application would be in season for something else. So, yeah.
Depends.
Yeah, it depends. I'm sorry, but...
Thank you.
I'm sorry, we're running out of time. So let's again thank David for his good presentation.